Welcome back, everyone. So in this video we'll go over your lab topic 5, cellular respiration. So just like always, you will need to download your supplement directions and have those available while you work through pages 101 and through 111 in your lab manual. So let's take a look at lab topic 5. So throughout this lab topic, you will be able to describe the process of alcoholic fermentation and how to measure it. So we've been talking about alcoholic fermentation in the lecture portion. So this will give us an opportunity to actually do an experiment on it. Okay, so we're going to be working with yeast and basically doing a closed system, so there won't be oxygen present, basically causing the yeast to undergo alcoholic fermentation. And then we'll be measuring how much ATP is produced, or the rate of the reaction, using the outcome or the byproduct of carbon dioxide to indicate how quickly the reaction takes place. We will also, you should be able to explain the steps involved in aerobic respiration and how to measure it. So we'll do some videos to relate to that. And then we're going to get into, you started this last week with the scientific lab proposal. You're actually going to run the experiment that you proposed for the scientific lab report. So if you chose the variable temperature or salinity or pH, you'll actually run this experiment this week with the scientific method. So just as we discussed in lecture, we're going to go through one of the metabolic pathways, alcoholic fermentation, which is how cells can obtain ATP through cellular respiration. So alcoholic fermentation being one of those processes. So in this process of alcoholic fermentation, recall that glucose is used just like in aerobic respiration, but then the byproduct that is produced is ethanol, or otherwise known as alcohol, and carbon dioxide. And then ATP is produced, but only about 2 ATP are produced per glucose molecule. So it's not nearly as effective as aerobic respiration. So let me go ahead and take a look at our lab supplement. So you're going to be watching a video on how to set up the experiment at home. So you will need some items from home. So you'll need some clear bottles to work with, and some sugar and some balloons, okay? And then you'll need some stuff from your kit. Okay, so we do have a video on how to set up the experiment, and then some videos on basically what it looks like throughout the experiment, okay? And then, you know, how to graph the data and so forth. So the in-person lab, they use something called these fermentation tubes. So they filled these fermentation tubes with a yeast solution. So yeast are the actual organisms that were used in this experiment, and they're single-celled eukaryotes. So they are, you know, eukaryotic cell. They have, you know, nuclei. And this fermentation tube is set up where we have the organism, the yeast, and we include water. And then we also include corn syrup. So corn syrup is sugar, right? So it's basically high fructose corn syrup. So fructose is something we can use that's very similar to glucose, right? And we learned about in the end, I think it was the last video of our lecture, that fructose can give us the same outcome as glucose in our reactions. So the yeast, they will eat up the corn syrup, And in this case, we are sealing up the container. So the yeast solution with the corn syrup, they're pretty much all sealed up here. And then we incubate this fermentation tube in an incubator at body temperature. So it was like 37 degrees Celsius or 90 body temperature. And so the yeast, the idea is they're eating away at the corn syrup, and they're undergoing alcoholic fermentation. And so as they are undergoing alcoholic fermentation, they are producing carbon dioxide. So as they're producing carbon dioxide, little bubbles are forming in this little test tube part of the fermentation tube. And so we can actually measure how much of the bubbles are forming. And so the more bubbles are forming, we can actually take a measurement. And so the more bubbles are forming, the more alcoholic fermentation is taking place. Hence, the more ATP is being produced. Okay? And so this, in this particular experiment, you can see the contents of the fermentation tube. So you can see here what would be the independent variable. So the amount of corn syrup is the same through all the fermentation tubes. So the only thing that's changing is the amount of yeast. Okay, so fermentation tube number 1 has no yeast in it, so that would be the control tube. Whereas fermentation tube number 2 has less yeast than fermentation tube number 3. Okay, and so as you go through the videos, you can see what happens during the experiment, and you can see, which, you'll write down the results based on the videos, and you can see which tubes ended up with the highest rate of fermentation. And of course, we suspect that nothing will happen in test tube number 1 because there's no yeast in there. So if there's no yeast in there, there shouldn't be any fermentation taking place. And then once you're done watching the videos and you've inputted all of the data, then you're going to write down the information, the answers to the questions, and then you're going to graph the data. So think about again how you're going to graph the data. Where does the independent variable go and where does the dependent variable go? And think about what type of graph you're going to create. Should you create a line graph or a bar graph? So, think about how this is going to be set up, because that'll help you later on when you're doing your actual experiment, your independent experiment, and then how that's going to be set up for your actual, final scientific report later on. Okay? In the second activity, exercise 5.2, we get into aerobic respiration. So remember, aerobic respiration is a lot more complicated because we go through the whole process of glycolysis and then the citric acid cycle and then oxidative phosphorylation. And all of that produces anywhere, from 32 ATP to somewhere, sometimes you see 38 ATP from one glucose molecule. So this process, if we were to do this in a lab, we would use something called a colorimeter. And so, of course, we don't have that here. So you're going to watch a video showing how it's set up. And then basically, you're going to see how this process, as it goes through, the colorimeters are set up to basically look at the absorbance rate of color throughout the experiment. So as the substance, we actually add a compound, it's called DCPIP, to the solution, and this particular compound, it's a blue dye, and it is a receptor for protons and electrons. So if you think about it, when do protons and electrons get used? So they're getting used during oxidative phosphorylation. So at the beginning of the experiment, the solution will be this blue color. But as oxidative phosphorylation takes place, the electrons and protons start getting used up. And so the color will start to lighten over time. And so this colorimeter, which looks like that, it'll basically measure the percentage. So as the percentage starts to change, we'll notice that basically the substances will start getting lighter, and as it's getting lighter, we know that cellular respiration is taking place. Okay, so the darker color means the greater percentage of absorbed light will be recorded. As the substance gets lighter, less light is absorbed. Okay, so more light is transmitted. So you'll see the percentage change throughout the videos. So you'll just record the percentages, and then it'll be another graph that you will draw, and then basically in this table shows you exactly what the experiment includes. So we have 3 test tubes. Each test tube gets mitochondrial suspension, so we're using lima beans. Lima beans being, the seed of a lima bean plant, and there's plenty of mitochondria in there, so the powerhouses of the cells. We have the DCPIP, so that's the dye that's going to accept the hydrogen ions and the electrons. And then we have a buffer, and then we have something called succinate. So succinate is the substrate in this reaction. So the substrate, you can see, is basically what's changing in this reaction. Okay, so you'll take a look and see when we do this experiment how depending on the amount of succinate, will the color change, the rate of the reaction, will cellular respiration occur, based on the amount of this succinate presence. Okay, so will it occur more quickly, will it occur at a slower rate, and so forth. Okay, and so you can read more about that process. You can see how the succinate, it should react with the enzymes present in the lima bean to turn to fumarate, and you should see during that time the electrons released will turn the color of the DCPIP from blue to colorless, and so we should see a change. So we should see the more succinate presence should hopefully lead to more color change. Right? So read the directions. You'll need to indicate the role of each of the components in the mixture, so make sure you're reading everything thoroughly. So here's the succinate, the substrate, and then what the product will be, and then what each of the components are used for, what's the independent versus the dependent variable, and so forth. Okay, and you're actually not doing the experiment part, so you just need to watch the videos, and then you can write the results there. Okay, and then finally we get into designing your own experiment. So in the designing your own experiment, you should bring out your own proposal that you have submitted, and this is a good opportunity to go back into our laboratory resources. And in our laboratory resources, remember how I told you last week in our week 4 intro video to just watch the first 10 minutes because that just has to do with the lab proposal. But now is a good time to, continue watching maybe, the next 15-20 minutes of the video, because that shows you kind of how to set up your experiment and run your experiment. We also have some directions here, so lab 5 directions for the how to run your experiment. These should be the same as what's in your lab supplement. I believe they are the exact same ones, so you don't need to go back. But whichever experiment you are doing, make sure you are following the directions. Okay, so you're going to make your own yeast solution. Okay, make sure your yeast is fairly new. You bought it recently. You don't want to use like 10-year-old yeast that's been sitting in the back of the cupboard for 10 years because it gets old and then it's not going to perform very well. So get a new package. So we have you'll make a yeast solution and a sugar solution, so just table sugar. And then depending on which variable you chose, we have the effect of the temperature experiment, so here's your directions for that. We have the effect of pH levels, so here's your directions for that. And then the effect of salinity, so here's your directions for that. So which variable you chose, make sure you've kind of set up your experimental design based on those directions, okay? So you can kind of transfer that information and write out, what was your hypothesis, what was your dependent variable, what was your independent variable, and so forth, okay? You're going to go ahead and perform your individual experiment, okay? So think about how you're going to set up your data table. You want to go ahead and take pictures throughout your experiment, so make sure those are available so that when you do make up your scientific report later on, that you will have those available for your scientific report. So now is a good time to take pictures of your balloons and all that when they're inflating. So don't forget to make your observations, write down your all of those observations, all of the data. Take good notes of what's going on throughout the experiment, and then take lots of pictures throughout your experiment, just in case if you need anything for your scientific lab report later on. Okay, so that's all here. All right, and then let's see. So let's go back into our so you'll have your results. So you could just, create your own data table, and then, think about what you would include. So this would be your discussion part, so you can write those down. Okay, so that's all included there. All right. So once you've collected all of your data and completed your experiment and completed the rest of your pages in your lab manual, you can go ahead and just submit the pages that you wrote on to the lab pages submission link. Again, you don't have to submit all the pages, just the ones that you wrote on. Don't forget to take pictures of your experiment while you go along, including a picture of yourself performing the experiment. You don't need to submit those pictures with the lab pages this week. Those will go in your scientific lab report. And your rough draft for your lab report is not due until next week, but you do have a midterm exam next week, so you might want to start working on the scientific lab report this week if possible. Or at least when you're writing up your experimental design, just make sure you're very detailed. Okay, so when you're doing your design, your experiment, make sure you're writing this information down in as much detail as possible. So what were all of your controls? Write down the method is as completely as possible. Write down the results as completely as possible so that you have that information there for when you write the lab report. And if you do want to get a head start on your lab report, we do have that available. If we go back into our Moodle page, sorry, I have a cat that's being a pest right now. If you can hear him in the background, he's being annoying. But if you go under laboratory resources in our Moodle page, you can go ahead and, watch, again, I told you to watch week 5, but you can go ahead and watch week 6 starting at minute 15 or 17, excuse me, and download the PowerPoint slides, as well as review the lab report directions. Okay, so all of that information is there for you if you'd like that head start. I recommend at least, maybe starting to write the method section. So since you are doing the experiment, write down exactly what you're doing, just so that you have that available, and then go ahead and write that paragraph out for your report. Okay? So as far as sorry, I just got kicked out. So as far as what is due for the lab, we have our lab pages that is due on Sunday, as well as your reviewing your knowledge. Okay, so if you have any questions, feel free to reach out.